this afternoon for this fantastically exciting lecture from Jennifer Doudna. I'm director of the Wellcome Center for Human Genetics. The Center for Personalized Medicine is part of that with St. Anne's College and Helen King. Together, we chair the steering group of the CPM. And without further ado, there's an introduction from the Stanley Ho Foundation, who support this lectureship.
Hello, and welcome to the Oxford Center for Personalized Medicine lecture by Professor Jennifer Doudna in honor of the memory of Dr. Stanley Ho. I am Ian Hewan, a trustee of the Dr. Stanley Ho Medical Development Foundation and CEO of the Atorum Group, a NASDAQ-listed biotech company. The foundation aims to enhance the quality of medical service and delivery in Macau, Hong Kong, and other regions of China, and enters into academic partnerships with relevant worldwide organizations to achieve this. It is our pleasure to have supported the Oxford Center for Personalized Medicine since 2013. The University of Oxford Center for Personalized Medicine is based at St. Anne's College and the Wellcome Center for Human Genetics, UK. Its principal aim is to engage a wide range of interested people, including academics, clinicians, students, patient groups, and the public, with personalized medicine. Dr. Ho's support for the center marks his vision and commitment to help transfer knowledge of personalized medicine to healthcare professionals and students worldwide. Dr. Stanley Ho passed away in Hong Kong at the age of 98 on May 26, 2020. He was not only an extraordinary leader, but also a great philanthropist. His legacy is his extensive altruistic efforts from disaster relief to epidemic control, having helped shape the communities and lives of many who have been touched by his generosity. I have lost a lifelong mentor. The foundation and I will honor Dr. Ho's memory by continuing his spirit of serving our communities. It gives me great pleasure to introduce tonight's speaker, Professor Jennifer Doudna. She's the Li Ka-shing Chancellor Chair Professor in the Department of Chemistry and the Department of Molecular and Cell Biology at the University of California, Berkeley. Professor Doudna has been a leading figure in the CRISPR revolution for her fundamental work and leadership in developing CRISPR-mediated genome editing. In 2012, Professor Doudna and Emmanuelle Charpentier were the first to propose the CRISPR Cas9 could be used for programmable editing of genomes, which is now considered one of the most significant discoveries in the history of biology. They were awarded the 2020 Nobel Prize in Chemistry for their pioneering work. Professor Doudna, it is my pleasure to welcome you as the inaugural Oxford CPM's Dr. Stanley Ho Memorial Lecturer.
It's a great pleasure to be here virtually. I am delighted to be part of this extraordinary lectureship and to be the inaugural lecturer. It's really incredible legacy of Dr. Stanley Ho, and I'm delighted to honor that. I'm going to share the slides. And I wanted to start off by, first of all, just acknowledging Oxford and thanking them for organizing this as a virtual lecture. It was planned originally to be, of course, at Oxford, and we hope that will be possible again in the not too distant future. But in the meantime, I'm delighted to share with you some of the science that we've been doing in the laboratory. And I thought I would break the talk into three parts. I want to tell you first a bit about the backstory of CRISPR. It's kind of an interesting example of the serendipity of small science, really, in discovering something completely unexpected in nature and then how it could be harnessed for as a technology for genome editing. And that's what I'll talk about in the second part. And then in the rest of the talk, I want to tell you a bit about some of the research we're currently doing to understand how the CRISPR technology works, and importantly, how it can be deployed for clinical use. And I'm excited to say that there are already clinical trials underway using CRISPR, and I'll say a bit about the status of that work as well. So, CRISPR is a tool that allows genomes to be edited in a programmable way. And I'll say a little bit about what that means. What you're looking at here is a representation of the CRISPR Cas9 protein in green with a molecule of RNA in yellow that provides the zip code, the molecular guide that allows this protein with its RNA to interact with double-stranded DNA and trigger double-stranded DNA cutting. And I have a video that illustrates exactly how this works. So we have Cas9 with its guide RNA engaged with a molecule of DNA. And if you watch, you'll see the DNA get cut. And so this is a system that is used in bacteria as a programmable adaptive immune system that allows bacteria to fight infection. And that's in fact how the technology was identified in the first place, was by a small set of scientists who were studying this bacterial immune system to try to understand how it worked. And so going back to the mid-2000s, there was some interesting suggestive research, primarily from bioinformaticians at the time, including Francisco Mojica in Spain, who identified DNA sequences in bacteria that were adapting in real time as bacteria became exposed to bacteriophage or viruses. And this was a tip-off that there might be a mechanism by which these bacteria could store genetic information from viruses in their own genomes and then somehow use it to protect cells from future infection. So this is a video that illustrates how we now understand that CRISPR works. And so you can see bacteria growing, say in a biofilm, and upon infection by a phage, if the bacterium has a CRISPR sequence in the genome, it can acquire a new sequence of viral DNA and integrate it into this special place in the genome where each viral sequence is inserted between a repetitive DNA element. The cell makes an RNA copy of that CRISPR sequence. The RNA is processed into shorter bits that each include a sequence derived from a virus. And then together with a second RNA called tracer and the protein Cas9, this RNA-guided surveillance complex is able to search the cell looking for a DNA sequence that has a matching sequence to the RNA guide. And when that match occurs, the protein unwinds the DNA and cuts it, allowing the DNA to be degraded in bacteria. So this is a terrific way for these prokaryotic organisms to acquire immunity in real time as they become infected with phage. So the biology here is very interesting. And then as my collaborator, Emmanuelle Charpentier, and I were investigating the mechanism of this process, it became clear that that this double-stranded DNA cutting activity of CRISPR-Cas9 could be harnessed in a different way in plant and animal and human cells because of the way that our cells deal with double-stranded DNA breaks. And that's shown here. So in eukaryotic cells, when DNA double-stranded breaks occur, the cell can recognize the break and repair it. And in the process, introduce a small disruption to a DNA sequence or integrate a new section of DNA that incorporates new genetic material at the site of the break. And this was research that had been going on for a couple of decades before CRISPR came along, and many scientists had recognized that by introducing a double-stranded break in a genome, for example, in a human genome, that it was possible to change that DNA sequence during this process of repair. However, the challenge was how to introduce a targeted double-stranded DNA break, something that is obviously non-trivial, there were earlier technologies for doing this that relied on programmable proteins like talons and zinc finger nucleases. However, these proteins took effort to engineer and had to be redesigned for every DNA edit. And the amazing thing about CRISPR is that it's easily reprogrammed by simply changing the guide RNA. So here we're watching a video that illustrates the CRISPR Cas9 protein with its guide entering the nucleus of a eukaryotic cell, and then surveying the sequence to identify a sequence that matches the RNA guide. And as you can see, this triggers DNA unwinding locally. It allows the two chemical active sites of the Cas9 protein to cut the DNA double helix precisely, just like you would cut a rope. And then those broken ends of DNA are handed off to repair enzymes in the cell that can fix the break by introducing a small change in the sequence, like in this example, or by integrating a new piece of DNA if there's a DNA template available in the cell. And so when Emmanuelle Charpentier and I published this work in 2012, it was very quickly adopted by multiple labs around the world. And now, 8 years later, has actually gotten to a stage where, in addition to being used widely for research, there are multiple opportunities for clinical applications that I'll discuss briefly. And so, you know, the one thing to mention here is that we're focusing on applications in medicine, but the exciting thing about CRISPR is that it's cross-cutting across essentially all areas of biology. And so over the last 8 years, there's been incredible advances in basic research, as well as both medicine and agriculture, that have relied on this fundamental ability to manipulate DNA in a precise and positional way in cells. And it's really opened the door to accessing the information in the human genome and in other available genomes in a way that up until that point had been difficult, or in some cases not really possible. So in clinical medicine, I wanted to say a little bit about some of the incredible opportunities that are coming up for doing editing that will have an impact on patients. And I think the vast majority of this type of editing involves somatic cell editing, where cells from a patient can be corrected using a CRISPR-based approach, and then used in those patients to correct their disease-causing mutation. And this is an example in the case of sickle cell disease, where the mutation that causes sickle cell disease has been well known for decades. And it results from a single base pair change in the human beta globin gene that gives rise to a mutant form of hemoglobin that causes the distinctive sickled shape of these cells and leads to a number of very devastating physiological effects on patients, including organ damage and great pain and very difficult disease to manage. And it became clear really very early in the whole development of the CRISPR technology that this approach of CRISPR could be used to correct this mutation directly, or to activate a secondary gene such as fetal hemoglobin in these affected cells that would mitigate the effects of the disease. And so that work, which began in culture, cells that were being cultured in the laboratory, has now progressed to a point where it's being actually applied in patients. And this is a great example of the first US patient to receive a CRISPR therapy for her sickle cell disease, Victoria Gray, who has made headlines over the last year and a half because she's done very well with this therapy. And so there's been a lot of excitement about the potential for this treatment to be truly impactful in patients that are suffering from not only this disease, but many other rare diseases that would otherwise typically be unaddressed and very difficult to either treat, or in some cases even to diagnose. And CRISPR can in principle do both. And so one of the things that I've been working on at the Innovative Genomics Institute is how we think about a therapy like this from the standpoint of access, because although the technology is exciting and we can see the potential to have an impact on people's lives, I think that to make sure that this kind of therapy in the future is affordable to people that need it is paramount. And so that's one of the things that I'm doing currently, is to really work on how we can make CRISPR into a technology that is accessible to the largest number of people. And just for example, this current therapy for CRISPR costs north of $1 million US dollars per patient. So it's clearly at a price point right now that would not be accessible to people around the world that would potentially benefit from it. Another aspect of CRISPR that I want to briefly mention here is the ethical use of a technology that in principle gives us the power to rewrite the human genetic code in a way that could impact future generations. And so one of the things that I've spent the last several years participating in are global conversations about the ethical use of CRISPR. And in particular, this comes up when we think about applications in the human germline, which means applying CRISPR in human embryos or eggs or sperm in a way that would create heritable changes in the human genome. Something that has been widely discussed and debated, and there's a series of reports that have been released, including the one shown here that was the result of an international committee led by the national academies in the US and the Royal Society in the UK. So I think this is a very important topic to continue discussing and to maintain transparency around its use, because there's no question that this type of research certainly is going forward. And I think in the future, there may be opportunities to use CRISPR in this fashion, but we have to make sure that that work proceeds with appropriate guidelines and responsibility. And so I want to just, in the last part of the lecture, I want to turn to what is currently going on in the research lab to address some of the ongoing challenges of CRISPR, as well as how it can be used to address even the current coronavirus pandemic. And I want to really talk to you briefly about three different questions that we've been posing from a research perspective regarding the accuracy and the efficiency of using CRISPR for any application, but primarily thinking about it in terms of applications in clinical medicine. And the first question is, how do we ensure the precision of editing? And so we are working on this in a number of different ways, but the short story I want to tell you today has been the result of a terrific collaboration with the lab of David Liu at the Broad Institute at Harvard that has involved investigating the activities of engineered forms of CRISPR-Cas9 that are capable of chemically editing an individual or a set of nucleotides in DNA that collectively are called base editors. And just as an example, and this is a figure taken from a 2017 publication from the Liu lab. This shows a collection of pathogenic human single nucleotide polymorphisms. And out of the total that were categorized here, about half of them could be corrected if it were possible to convert an AT base pair in DNA to a GC pair. And of course, doing that site specifically is a tall order. And yet the CRISPR technology is in principle capable of this kind of precision chemical modification if it can be harnessed in the right way. And I'll show you very briefly the approach that the Liu lab and the Kondo lab and others have taken to creating base editors from a CRISPR platform. The idea was to take the CRISPR-Cas9 protein that I showed you before and make a connection, a protein linkage to a domain of an enzyme over here in red that is capable of converting adenosines in DNA to inosines, which can then be converted further to guanosines when the cell goes through a round of DNA replication and DNA repair. And so this was kind of the, the schematic of, wouldn't it be great if, this could happen? And the idea here was that Cas9, because of its mechanism that as I showed you, it's a protein that uses the guide RNA to trigger DNA unwinding. It's catalyzed by the Cas9 protein, and then in the natural setting, DNA cutting. And so in this instance, what the idea was to remove the cutting ability of Cas9 or turn it into a nickase, so it could only cut one strand of the DNA in a fashion that would tell the cell which strand of the DNA was the correct strand for DNA repair, and then allow this DNA unwinding capability to expose a sequence of DNA for editing at a particular position by this editor domain that was appended to Cas9. So that all sounds great. The challenge was that there were many challenges, but one of them is that there's no natural enzyme in nature that has this chemical capability of modifying a single-stranded DNA to convert an adenosine to anything really, but certainly not this pathway here. However, there is a domain, there is an enzyme that's well known in bacteria that can do this chemistry in RNA. And this is a tRNA modifying protein called TAD-A that functions as a dimer. Its structure is well known. And so the strategy of the Liu and the Kondo labs was to use directed evolution in the laboratory to introduce mutations into this TAD-A protein and then select for enzymes that would have the ability to do the kind of editing that I showed on the last slide. And through a number of rounds of selection, there were a series of these types of chimeric editing proteins that were appended to Cas9 that were evolved in this kind of laboratory setting to do adenosine to inosine and then guanosine conversion in cells. And so this was great from a sort of an editing, technology perspective. But the question was how to turn this ultimately into a tool that would be precise enough and effective enough that it would actually be useful in a clinical setting. And so this is where we started to work with the Liu lab and doing what we like to do, which is diving into the molecular mechanisms of enzymes. We did some time course measurements in the laboratory with purified versions of these different generations of editing proteins. And we found that most of them were very, very slow enzymes. So this is looking at a time course here. This is a 1 hour incubation of these purified editing proteins, incubating with double-stranded DNA and looking at the chemistry that's shown here that can be detected very nicely with using an endonuclease cleavage assay. And what was striking was that there was one editing protein that hadn't at the time been very well characterized from the Liu lab they called it ABE8E, which stands for A base editor, eighth edition, I guess. And this protein had markedly different kinetics in vitro compared to all of these other A base editors. So we scratched our heads and said, I wonder why that is. And we dug in to start figuring out what might be special about this particular version of the A base editor. Why was it so good at modifying adenosine in this kind of a setting? And so one of the things that we wanted to do was to try to catch it in the act of actually editing DNA. And so this led to a wonderful collaboration, three-way collaboration with the lab of Peter Beal at UC Davis, who has a long history of working on the TAD-A protein from bacteria, and they had done structural biology on this and had also come up with a very nice chemical strategy for trapping the TAD-A enzyme in the act of editing tRNA. And they could do this by substituting a nucleotide analog called 8-azanebulerin, which is shown here, at the position of editing in a tRNA substrate. And this would lead to a partial reaction in the presence of TAD-A that could not carry on to the rest of the, to actually complete the chemical conversion of adenosine to inosine. And so the idea was to synthesize DNA molecules that would contain azonebulerin at different positions in this part of the DNA that we knew would become exposed as a single strand during Cas9 binding, and then see if we could actually capture the editing domain of this ABE8E protein in the act of editing that position. And so this was work done by a postdoc in my lab, Audrey Lapinate, and Cody Palumbo, a student with Pete Beal. And after summarizing 2.5 years of work, they eventually did find a position modification in the DNA that succeeded in this kind of trapping. And furthermore, we were able to capture this and visualize it using cryo-electron microscopy together. And this was work done by a second postdoc in the lab, Gavin Knott. So Gavin and Audrey worked together to do the structural biology on this protein. And I'll show you just a very short animation of this. So you're looking at the Cas9 protein in white. The red and pink are the TAD-A editing protein bound to DNA. And hopefully you can see that the protein is holding on to its guide RNA in magenta. The DNA is literally unwound as it traverses the protein. And this is the important part. This is really the action area of this editor, is the loop of DNA that enters the active site of the TAD-A enzyme. And I won't show you in detail here, but we could actually observe that the 8 mutations that occur in the TAD-A version, this 8th edition version of the A base editor, are all clustered in the region of TAD-A that bind to DNA. And so it really was very clear that this enzyme has become adapted for a DNA substrate. It now precludes editing RNA. It doesn't edit RNA very well anymore, but it's terrific at editing this kind of looped out single-stranded DNA molecule. And in fact, it's almost too good, because we did find, and I won't show you these data, but they're now published, that this protein is actually also editing other positions along the DNA, probably as a result of the search mechanism of Cas9 as it moves along DNA looking for a target sequence to bind with its guide RNA. And so we think that there actually may be a happy medium to be still discovered that will identify a version of this modified editing domain that will be still fast and effective at editing DNA, but not so fast that it is able to capture transiently melted DNA during the Cas9 search process. So this is an illustration of how understanding the molecular mechanism of these types of proteins can in fact guide the direction and development of a technology like this. I also want to point out that in nature, there are a number of alternative proteins to the Cas9 protein that Emmanuelle Charpentier and I first investigated. And we've been interested in finding these and understanding how they work. And this has been a long time collaboration with a colleague of mine at Berkeley, Jillian Banfield, who researches bacteria primarily growing in various environmental settings. And she's one of the international leaders in metagenomic sequencing, which means collecting bacterial samples, sequencing all of the DNA in the samples, and then reassembling genomes from those data to identify what organisms might have been growing in those settings. And of course, in the context of this type of work, they also identify bacteriophage that are infecting these organisms. And that was actually she was one of the early labs to identify CRISPR sequences based on that type of work. And so very recently, we've been working with her laboratory on a fascinating subset of CRISPR-Cas systems that are found only in phage. So one of the surprises here is that an anti-phage mechanism, of course, has been picked up by phage, and we think is being utilized by some phage, at least, to target their phage competitors. And one of these proteins is a protein that we call Cas9 because this is a protein that is about half the size of the original CRISPR-Cas protein. It's about a 70 kilodalton protein. And it's in a very small CRISPR locus. It has just a single, this one gene together with a CRISPR array of sequences. And it has just a short guide RNA that it utilizes for DNA targeting. And so in work that was done with Bassem Al-Shayeb, a graduate student, and Patrick Pausch, a postdoc, we were able to start characterizing this CRISPR-Cas9 system. We found that this system is in fact the smallest so far that's been identified in terms of its total molecular weight compared to any of these other CRISPR-Cas type systems. And yet, remarkably, it's still capable of not only targeted double-stranded DNA cutting, but also of inducing genome editing in human cells using a very simple assay that where we can identify and quickly sort cells that have been edited based on targeting of a green fluorescent protein encoding gene in these cells. And this is just showing some quantification of different guide RNAs that work in these cells. And so we've been, since this work was initiated, we've been continuing to investigate how this very small protein is nonetheless capable of DNA unwinding, DNA cutting, and triggering this kind of editing in eukaryotic cells. And so recently, Patrick Pausch and another postdoc in the lab, Kasia Selczek, have been using cryo-electron microscopy to capture the Cas9 protein in different structural states. And I'll just show you one image here of the protein. So we know that we now know from this work that the protein hugs the nucleic acid very, very tightly. It sort of wraps around the guide RNA, which is in orange in this image, and the targeted DNA. And we can see now in detail how the DNA is recognized by this protein. We have some important clues about how DNA unwinding is triggered in these enzymes, these very small proteins. And we also have some data showing that these proteins are naturally inhibited from cutting DNA randomly by a structure that, when removed, makes these enzymes even more active than they are in nature. So this is something we're currently exploring, and I think is, again, illustrates the value of having this kind of mechanistic and structural data that we can use to understand the natural functions of proteins like this, and then engineer them to do new things as needed for technology development. And then finally, I wanted to briefly address this question: how can CRISPR-Cas editing enzymes be delivered to specific cell types? I think this is the big current bottleneck, honestly, in the CRISPR field, is really figuring out how to do this. I don't think there's likely to be a one-size-fits-all answer to it, but clearly having better ways to deliver these editors into tissues and cell types selectively is going to be key. And just as an example, going back to the story of Victoria Gray and other patients who have been treated using CRISPR, right now that treatment requires bone marrow transplantation. And in those studies so far, the primary negative health impacts on those patients have been a result of the bone marrow transplant rather than apparently anything to do with the genome editing. So it just speaks to the importance of figuring out safe ways to deliver these types of molecules in different settings. And so what we're doing to address this right now is really thinking about ways that we can package the CRISPR protein with its guide RNA for delivery. And I wanted to just point out this paper from last year from Carol June's lab that was, again, one of the highlights of last year in terms of developments in the CRISPR field, because it really showed the potential to use CRISPR engineered T cells in patients who are suffering from cancer, and highlights the future potential of this kind of a strategy. And as shown in this image, this type of T cell engineering today involves, again, ex vivo editing. So the cells are removed from a patient, the editing goes on in the lab, and then the edited cells are returned to the patient. And so we've been thinking about this and wondering if there might be an alternative in the future. Wouldn't it be great if you didn't have to do all of this and you had a way to provide a one and done injection, or who knows, maybe someday even a pill that could be taken that would deliver the editors just to the cells where editing is necessary. And so this is a project being conducted by Jenny Hamilton and Abby Stahl, two postdocs in the lab, and Connor Sushida, a bioengineering graduate student. And the approach they are taking is to use virus-like particles as nice little packages for capturing assembled CRISPR-Cas9 proteins with guide RNAs. And the important thing here is that we can take advantage of the glycoproteins on the capsid surface for delivery into cells, because this is effectively the viral infection mechanism. Importantly here, these virus-like particles have been gutted of genes that would allow virus replication. So they're not infectious in that sense, but they're able to transduce particular cell types and dump their contents into the cells. And so the way that we're doing this, and this has been a terrific collaboration with some additional scientists at UCSF in the lab of Alex Marson, in particular David Nyan and Brian Shai, two MD PhDs who are focused on T-cell engineering for immunology applications. And the strategy here is to use it's kind of a 1-2 punch, where we have Cas9 protein RNAs. So these are ribonucleoproteins, or RNPs, that target genes important for this type of a T-cell engineering approach, together with a lentiviral genome that encodes a second a piece of DNA that can target another gene, in this case, in a anti-CD19, which is cell surface receptor, chimeric antigen receptor protein, gene encoding its protein. And so the idea here is to use the CRISPR-Cas9 system to target and knock out particular genes, and at the same time allow integration of a new gene that can create that chimeric antigen receptor, so useful for T-cell therapies. And so this is very briefly a summary of the strategy. And so what we can do is to encode, in addition to genes important for creating these lentiviral capsids, we actually encode a chimeric GAG protein that's been fused to Cas9. And this is important because when these plasmids are transfected into cells, they create these kinds of particles in which the Cas9 protein is anchored to the capsid. So this ensures accurate or efficient encapsulation, together with guide RNA. And then there's a proteolytic cleavage site that allows clipping of this assembled Cas9 off of the interior surface, so that these are released inside the capsid. And so I'll just show you a couple of data points here. And so that we're able to use this type of a strategy to get pretty robust expression of this chimeric antigen receptor in these targeted cells. And we can quantify this in different ways. So this is showing quantification of two different types of T cells, these CD4 positive cells and CD8 positive cells shown here. And importantly, we can also engineer the capsid of these virus-like particles to target just one type of T cell. And so this is an experiment in which it was possible to selectively target just the CD4 positive cells in a mixture of cells that also contained CD8 positive cells. And so we think this is a strategy that in the future has real potential to allow targeted editing of just those cells that will be clinically beneficial in a patient, while leaving all the bystanders, standard cells unmodified. And then finally, just wanted to mention also that we have a lot of interest in applying this strategy of protein RNA delivery for genome editing in the brain. And that sounds like a high bar, and it certainly is. But I think there's such an important unmet medical need right now for treating patients that have neurodegenerative disease that we've been very interested in this. And This is actually a project that started several years ago in the lab with the work of a former postdoc, Brett Stahl, who came in and was able to show that he could engineer Cas9 with peptides that were appended to the protein surface that allow cell penetration. And so this was a kind of a direct protein delivery strategy where these engineered Cas9 and guide RNAs are injected across the blood-brain barrier. This is an experiment conducted in a mouse using a reporter mouse where editing leads to cells that are red. And so it's very easy to see cells that become edited. And you can see a nice kind of dose response here that the higher the amount of injected engineered Cas9, the more editing is observed. And since then, we've been able to improve steadily improve the efficiency of this and achieve really quite decent levels of editing again in the mouse. We're still investigating how this would work in a larger brain, like in a larger sort of animal brain. But I think the principle here is that we think that this will be a useful strategy for, especially for targeted delivery, where there's a desire to edit cells in just one region of the brain. For example, the kind of therapy that might be useful for treating something like Parkinson's disease. And then just one final slide, I wanted to mention CRISPR and coronavirus. It's a little bit hard to, sort of imagine the complexities of the past year for everybody really around the world. But when the pandemic was just getting started in the US just about a year ago, we realized at the Innovative Genomics Institute that there were some really important things that we could do as an institute to address this emergency. And one of the things we did was to set up a clinical testing lab, just using a standard PCR-based technology. But that's been incredibly important because we're now a clinically approved laboratory that is able to test all of the samples from people across our university campus, as well as we work with a number of healthcare partners in Northern California that has allowed us to provide regular testing for people that would otherwise not have access to it. For example, folks that are unsheltered, folks that are first responders of various kinds. We've worked with a number of our fire departments and police departments to provide them with regular testing. And in the process of doing that, we also realized that CRISPR could have an important role to play in testing and diagnostics as well, based on some interesting chemistry that emerged from our lab several years ago now, starting with a graduate student, Alexandra Seletsky, who first showed that you could use the activities of some proteins, in particular a type of protein called CRISPR Cas9 that is naturally an RNA targeting form of CRISPR, to report on the presence of a sequence. And very briefly, the way this chemistry works is that what Alexandra Seletsky showed is that these proteins naturally have an ability to recognize RNA with an RNA-guided mechanism, so very analogous to what I showed you for Cas9, except that in this case, recognition of a sequence turns on a non-specific RNA cutting activity that will cut any single-stranded RNA in the vicinity of this enzyme. And so she realized, this was also published at a similar time by Gutenberg and Feng Zhang. But what Alexandra recognized was that she could use this activity to report on the presence of a sequence if she had a quenched fluorophore pair of dyes that were coupled to single-stranded RNA. And so this this is a reaction then that upon activation by recognizing a target, this enzyme starts cutting the reporter and releasing fluorescent signal that can be easily detected. And this fundamental strategy has now become widely adopted by a number of labs and companies. There are a second class of enzymes called CRISPR Cas12 that have a similar capability except for DNA recognition. So they have a DNA cutting activity that gets turned on upon binding to DNA sequences. And so this is a great way to use CRISPR as a diagnostic tool. And so we've been recently, this is a very recent publication that is from the lab of Melanie Ott at the Gladstone, another one of our longtime collaborators, to use CRISPR as a tool for detecting viruses. And you may know the coronavirus is an RNA virus. So having a technology that can directly detect RNA without having to go through a DNA step where you have to copy the RNA into DNA has some real advantages in terms of quantification as well as speed of the test. And so that work is very much, very actively under development. And in fact, we're excited that at our clinical testing lab, we're just about to start using a CRISPR-based laboratory test for coronavirus that will begin. We're one of the beta test sites for a local company that's been developing the technology, and we'll see how it works with our robotic pipeline in the clinical lab. So I just want to wrap up there and just point out that, I think continued mechanistic dissection of these systems will guide the further development of fast and accurate genome editing proteins. We think that continued discovery of new genome editors, including those that are very small and compact, will give us new strategies clinically, especially for delivery. And finally, that figuring out better ways to deliver these proteins, especially in situ, will be fundamental to the continued advance of this as a real clinical therapy for lots of other diseases in the future. I want to thank a great team. This is obviously a pre-pandemic photograph, including all of my, I think I had 15 undergraduate students that were working in the lab that summer. So this is showing the entire team here. And I just want to give a shout out to our original work with Emmanuelle Charpentier, with Martin Yinek and Chris Chylinski, who did the work on Cas9 originally, our labs at Berkeley and Gladstone that are doing the research that I talked about today from our group, as well as our collaborators, Alex Marson, Melanie Ott, and also Bruce Conklin at the Gladstone Institutes. And of course, all of this is made possible by support from these agencies here. Thank you very much. And of course, I'll be delighted to address questions that you might have.
Thank you, Jennifer. Wow. So humbling and inspiring all at once, illustrating fantastically the combination of biochemistry and structural biology coming through to that understanding to lead to the advance. We've got quite a few questions, and we've got a good 10 minutes. So many of them that you came in early, you've asked, you've already answered in your presentation about how might it help in cancer. There's one of the common questions. What would be your main concern, if any, going forward for the future of using CRISPR technology?
Well, I guess I have two. One is that I think that, as exciting as it's been to see the rapid advances, I think there's also needs to be appropriate caution with a new technology. So we want to make sure that as these, especially clinical trials, advance, that there's appropriate sort of restraint, I guess, to make sure that we don't get ahead of the technology. I think so far that's been true, and I've been pleased, and I hope that continues. And the other thing I'll mention briefly that I do think about, and I feel some angst about, is the accessibility, as I mentioned. I think that for this technology to really become impactful for people who need it, it will need to really come down in cost, and it needs to become much more accessible. And that's going to require not only delivery approaches, but also ensuring that, of course, it's accurate and safe and effective, and that it can be tested safely for many different applications, which is another challenge. I didn't talk about it, but if you think about it, also is an interesting challenge for a technology like this.
Yeah, one of the questions was how do you direct the therapeutic to specific cells? And again, you addressed that. There's a lot of work to be done there, but it looks as if it started and will be really important to get cell specificity on delivery.
Yeah, absolutely. Yeah.
And also, I think the approach taken again illustrated how sophisticated and how much thought goes into engineering to get that specificity. That was really impressive. one of the, I know microbiome sequencing has taken off, BGI, the Million Human Microbiome Project. But of course, what's waiting in the wings is the characterization of probably of millions of more phage. So it again, you mentioned this, it seems almost unlimited functionality in all the phage that we've yet to discover. It's
kind of mind-blowing, isn't it? I mean,
it is mind-blowing.
Yeah, I hear this regularly from my colleague Jillian Banfield, who does this work. You know, she's always telling me, Jennifer, we're just at the very, very tip of the iceberg, and the iceberg is huge. So
yeah, and there was one question about is there any possibility of anti-drug antibody responses, so the host immune system reacts against one of the proteins that's been brought in?
Yeah, that's a very important question, and the answer is absolutely yes. And there are several publications already that show the potential for that. Of course, we're using a foreign protein here, right? So you could imagine that could be immunogenic, and not only that, but the most commonly used form of CRISPR, the CRISPR Cas9 protein, comes from a pathogenic bacterium that many of us already have antibodies to, and so you could imagine that could also be problematic in people. It's not been demonstrated to be the case, but it's something that I think requires careful attention. We have a collaboration with a couple of immunology labs at Berkeley who have been looking at this question, and so they have, working in a mouse model, they have found that yes, indeed, that not surprisingly, that these bacterial proteins are immunogenic, but also that they can identify the immunogenic epitopes on the surface of these proteins, and they've been starting to engineer them to be, to be sort of humanized in ways that people have done effectively for antibodies. So I think that there may be opportunities there, and then also I didn't mention it, but one of our other motivations for studying these alternative CRISPR-Cas systems is that if they come from phage that have never infect bacteria that have nothing to do with human physiology, then we think that they may actually be intrinsically safer because they won't be pre-existing antibodies in people for this.
Yeah, and the same group published a very nice science paper recently. The same group that developed in Germany developed the Pfizer messenger RNA. And the title of the paper is a non-inflammatory messenger RNA to a self-antigen. So in parallel, there'll be better ways of tolerizing you to the key proteins that you guys and others develop. My colleague Peter Donnelly, who was the former director of the Wellcome Centre here in Oxford and also co-chair of the CPM, he's asked a question. The human genome's a big one. How does the Cas9 system find its sequence?
That is such a great question. I would love to know the answer to that. Yeah, I mean, that's, I actually just hired a new postdoc from the lab of Xiaowei Zhuang, one of the, really top imaging laboratories who does single molecule imaging. We're hoping to start to dissect this by doing some live cell imaging. But right now, there are a handful of publications about this. And, there's been some work, we did some work a few years ago with the lab of Bob Tejan on this topic, where it's possible to put a fluorescent label on Cas9 and then kind of watch its, movements around cell nuclei. But those experiments so far have just kind of told us the outer boundaries of, how this works. We don't know yet in detail how it actually finds a target. And I'll just leave you with an interesting conundrum, and that is that there are a lot of potential sites of targeting for Cas9 in a cell, and if it spent too long on any one of those, you could imagine it would literally never ever have time to get to its true target. So this has to be a very, very fast protein, and understanding how that works, I think, is still really a fundamental aspect of the field.
Wow, and it looks as if with the latest technology, you can image it as it travels across the genome.
Yes, Yes, so I think there's a lot of opportunity there now with the breakthroughs in imaging technology for sure.
Yeah. So maybe going back to the very beginning, somebody's asked of the several Cas enzymes, 12, 6, 13, 9, how did you settle on 9?
That's another good one. Well, I have to give a shout out to Emmanuelle Charpentier here, because that work really initiated when she and I met at a conference, and she had been already investigating this CRISPR system in Streptococcus pyogenes, in an infectious bacterium that infects humans, and trying to figure out how its CRISPR system was working. And it used a Cas9 based system. And at that time, my lab had been doing work on other types of CRISPR proteins, but not that one. And she said, I think Cas9 is it's clearly very important genetically in these cells, but we don't know what it does biochemically. So we want to work together to figure it out. And that's how we got into it. But it was clear from already in the field at the time that there was something very, very interesting about a single protein, Cas9, that had this RNA targeting capability where it could recognize a DNA sequence and cut it. It's just that nobody understood the mechanism, and that's where we got involved in it.
Yeah, and related to that, I guess, I mean, is there a Cas24 when you characterize, presumably there's millions of bacteria that remain to be found and sequenced?
Yeah, well, there's definitely a lot of diversity of CRISPR systems for sure. I think what's emerged so far in the field is that the Cas12 family of proteins that I mentioned it just very briefly, but that turns out to be quite a large superfamily of different flavors of those Cas12 proteins. So that's been kind of expanding over time. Whether there'll be is there going to be a Cas15, 16, 17, 18, 24, 58? I don't know. My guess right now is probably not, but if they are there, they'll be rare, I think. But again, I got to remember that we're at the tip of this huge iceberg of bacteria. So what else is out there? Yeah, we don't know yet.
And of course, another impressive feature was that evolution in the laboratory, searching for variants that do what you want of the existing Cas genes. I mean, that also was incredible. So we've got through the questions. I'll ask you one more. When you must give this seminar a lot, what's the toughest question you're asked?
Well, I guess I would say two tough ones are about ethics. So nobody asked in this seminar about editing human embryos, but that's one that's been on many people's mind. Do we have to worry about rogue use of CRISPR-Cas? And my answer there is yes, but I think that the potential of the technology is so huge and it's so important that we don't want to slow it down. We just want to make sure that we, I think the global scientific community needs to be making a strong stand about how we want to see the technology proceed, and I think that's been happening. So I'm when there's never I don't think there's ever a way to enforce global regulations. We have a hard time even doing that in particular countries. But I do think that having some clear criteria that are agreed to by authorities worldwide is really helpful. And that's what's been happening with that. The other tough question, I guess, is about patents and intellectual property. So nobody asked about that either.
Yeah, there is a question on that. Yeah.
Yeah, and I think the challenge there is that this is a technology that I think all of us I think would want to see adopted as widely as possible, right? And and developed as quickly as possible to be beneficial to the largest number of people. And the question is always how to do that, appropriately and to allow investors who might be developing commercial ventures to recover their investments, which is really the purpose of the patenting system. And so, I've really been pleased that the science, I think, of CRISPR has continued to develop quite quickly, independent of the ongoing patent disputes, which unfortunately I think those are gonna be with us for a while. They're gonna go on and on and on, and lawyers are gonna be the winners there for sure. But I think fortunately the science and the application of it continues apace, and so this is really key to keep in mind. Is that, when you read about the latest in the patent fights in the media, remember that labs worldwide are continuing to do their work, and this is not slowing any of it down.
All right, we've reached 6 o'clock UK time. Thank you very much indeed. Fantastic. Thanks.